All right, let's jump right in. Today, we're going to unpack some really fascinating research that suggests a potential link between a tiny piece of the SARS-CoV-2 virus, our immune system, and get this, proteins that are critical for brain development. It sounds complex, but we're going to break it down step by step. Okay, so right out of the gate, here's the core idea we're exploring today. Think of this slide as our roadmap. The hypothesis is that a specific little piece of the virus, a peptide, looks exactly like a sequence in one of our own human proteins. And because they're lookalikes, our immune system could get tricked into attacking our own cells, potentially creating a direct link to how our central nervous system functions. Let's dig into each part of this chain reaction. So, how in the world does something like this happen? Well, it all comes down to a scientific principle that's basically a case of mistaken identity for our immune system. Let's get into the science of what's known as molecular mimicry. At its heart, molecular mimicry is when our immune system just gets its wires crossed. It's trained to be an expert at hunting down and destroying foreign invaders. But every once in a while, a part of a virus or bacteria looks so much like one of our own proteins that the immune system can't tell them apart. It launches an attack on the virus, which is great, but then it keeps on attacking our own healthy cells. It's basically biological friendly fire. And here's the really wild part. It doesn't take much to cause this confusion. The research shows that a shared sequence of just 5 amino acids, those are the building blocks of proteins, can be enough to fool a T cell. This diagram here shows it perfectly. You see how that single T cell receptor can lock on to both the viral piece and our own self-peptide? That's the moment it happens. That's what triggers the cross-reactive autoimmune response. Okay, so we get the mechanism. Now let's play detective. What's the first piece of this puzzle? What's the specific part of the virus that's actually pulling the trigger? The viral peptide we're talking about is this 5 amino acid sequence, RLFRK. And its location is absolutely critical. It's not buried deep inside the virus somewhere. No, it's sitting right on the surface, on a part of the spike protein called the receptor binding motif. This is the very part of the virus that physically docks with our cells, which means it's right out in the open, perfectly positioned for our immune system to see it immediately. And this isn't just a guess. Research has confirmed that this whole area is antigen-rich. So, as this diagram shows, our immune system's scout cells, they're called antigen-presenting cells. They find this peptide, chop it up, and present it to our T cells. It's like the handshake that officially kicks off the entire adaptive immune response. The system is primed and ready to react to the specific piece. All right, we've identified the viral trigger. Now, let's meet the other side of this story, the unfortunate victims in this case of mistaken identity. Let's look at the human proteins that look just like that piece of the virus. The first human protein that has that identical RLFRK sequence is made by a gene called PPIP5K2. And this is where the first major connection clicks into place. If you look up this gene in the official government database, the National Center for Biotechnology Information, its summary literally states that it may be associated with autism spectrum disorder. So right away, we have a direct, documented link between our target protein and neurodevelopment. But that's not its only job. This protein is a serious multitasker. It has a second, incredibly important role. It's a key regulator of our body's #1 defense system against viruses, what's called the interferon response. So think about what this means. An autoimmune attack on this one protein could cause a double whammy of problems. First, as you see on the right, it could weaken our innate ability to fight off viruses. And second, it could mess with all the other critical jobs that this protein is supposed to be doing inside our cells. And if you're thinking, well, maybe that's just a 1 in a million coincidence, it's not. The evidence shows that the exact same RLFRK sequence pops up in a second human protein, one called 10M4. The pattern repeats. And just like the first target, the gene for 10M4 is also identified as a risk gene for autism. Its day job is super important for the nervous system. It's essential for axon myelination. Think of that like the rubber insulation around an electrical wire. Myelin wraps around our nerve fibers so that brain signals can travel super fast and without interference. This image just makes it crystal clear, doesn't it? On the left, a healthy neuron, all nicely insulated. On the right, you can see what might happen if there's an autoimmune attack. That myelin sheath starts to get damaged. This process is called demyelination, and it can really disrupt nerve signaling. It's also a feature that has been documented in some individuals with ASD. And believe it or not, there's even more. The evidence points to a third protein called POMGNT1. Now, this one doesn't have the exact same sequence, but it shares a larger, structurally similar piece of the spike protein. And its job? It's critical for neuronal migration, which is basically making sure that baby brain cells move to the right spot during development. As you can see from the diagram, when that goes wrong, it can cause some serious issues. Okay, let's take a quick pause. We've seen the viral trigger, and we've met the 3 human targets. Now it's time to put all of these pieces together and look at the whole cohesive story. This table is the perfect cheat sheet. In that first column, you've got the viral peptide. It's out in the open, and we know it gets the immune system's attention. And then you've got our 3 human targets, PPI5K2 and 10M4, both with the identical sequence and both linked to ASD risk. Plus POMGNT1, with its structural similarity and its vital role in building the brain. And here it is, the entire proposed pathway laid out in one image. You start on the left with the viral trigger. That kicks off an immune response. But because of molecular mimicry, that response gets confused and cross-reacts with our own human proteins that are essential for our immune defense, for insulating our nerves, and for brain development. The potential result? Dysregulation of the central nervous system, especially in people who might already be genetically vulnerable. So what's the big takeaway here? We have to be really precise. This convergence of evidence, the active viral peptide, the perfect mimicry in multiple proteins already linked to ASD, and the critical jobs those proteins do, it all establishes what the source material calls a robust and plausible candidate pathway. This is not definitive proof, but it is an incredibly compelling hypothesis that absolutely deserves more research. And that really brings us to our final thought. When you see a potential link like this between a common virus and these fundamental pathways for brain development, it really makes you wonder, how critical is it that we start exploring these environmental triggers, especially in people who might be genetically more susceptible to begin with? It's a huge question for the future of this kind of research.